My name is Natasha Ayoub, and I'm a PhD candidate with University of Waterloo. I'm investigating spatial and temporal movement patterns of Yukon River juvenile Chinook salmon. I'd like to acknowledge the traditional territories on which I conduct this research. I'm grateful to call these beautiful lands my home. The research focuses on the Yukon River watershed. The entire watershed encompasses close to 900,000 square kilometers, while the Canadian portion is around 255,000 square kilometers. One key component to this research is collecting water samples throughout the watershed. This includes taking notes, collecting samples, organizing the samples, shipping them to Waterloo, and then having them analyzed at the Environmental Isotope Lab at University of Waterloo. The water samples are analyzed for strontium isotopes, and each of the analysis is then placed on a map according to where it was collected on the site within the Canadian portion of the Yukon River. Another key component to this research project is the collection of otoliths. These are tiny calcified tissues that are found in a Chinook salmon's head. Once these otoliths have been removed from the Chinook salmon's head, they need to be cleaned thoroughly. They're then embedded in epoxy resin. Each otolith is then given a unique identifying code. Each dobby is inspected under a microscope to identify the nucleus. The nucleus is marked, and then the dobby is placed in a sectioning saw where we can get a sliver that exposes the nucleus as well as the annuli for the otolith. These slivers of otolith are then placed in epoxy resin discs, and the discs are then sanded and prepared for laser ablation. The discs must be free from any scratches or lines because the laser ablation machine is highly technical. The laser ablation machine then burns a line from the nucleus out the distal edge and results in analyzing the strontium levels. Seen here is an image of an overlay of the ablated otolith with the laser ablation analysis of the strontium ratios. This analysis is generated for each individual otolith, resulting in a unique pattern that demonstrates the movement of this juvenile Chinook salmon while in the freshwater environment. There are many components to this research project, but I'm working hard to analyze the data so that spatial and temporal movements of juvenile Chinook salmon in the Yukon River can be better understood. It's my hope that by better understanding these important juvenile movements while they're in the freshwater, this will help Yukon First Nation governments with planning for protection and conservation in areas that are critical to these juvenile salmon. I'd like to thank Wildlife Conservation Society Canada for the mentorship that they've provided throughout this program. I'd also like to thank my collaborators at Yukon First Nation Salmon Stewardship Alliance. And of course, the generous funding support of the Weston Family Foundation. This funding has allowed me to analyze the water samples as well as the otolith samples and to provide results for this important research project. Thank you very much.